kind of just arrived on the scene in genomics purely out of his own interest without any sort of formal background, and started to work with geneticists and genomicists purely out of his own interest and his own particular route. These days these things are a bit more formalised. People can go on special programs to get this kind of training, but Daniel did it all for himself, and that's the kind of guy he is. So he's currently at the Max Planck Institute for Molecular Genetics.
No, but it doesn't matter. Keep going.
Is that wrong?
Yeah, try.
Well, it was okay.
No, I told you it wasn't okay. Just keep going.
Okay. I know he moves around a lot, but
Yeah.
not quite that far. And his main interest is in evolutionary anthropology. So over to you, Daniel.
No, yeah. Okay. No, so I'm. So before I start my talk proper, I sort of want to make a comment on this, which I think is very relevant to this workshop, on the question of assessing community composition. Okay, and the kind of assumption that I would say that people have made in the talks is that in order to estimate community composition well, what you need to do is sort of assign reads ever more accurately. And I sort of want to show an antidote to this. This is just a human genetics project, and humans are very similar to each other. We share ancestry amongst all different groups, and we have this thing called chromosome painting. And we've made an artificial mix of an African and a East Asian chromosome. So this is the kind of simulated data. And if you try to paint it using other individuals who you're similar to, you get a very messy picture. But the point is that if you sort of take this kind of thing and averaging it over a whole genome, then you can sort of clean it up a lot, and you can make very accurate inferences. And the kind of this is the kind of the extremes of this. This is from the Peopling of the British Isles project. I'm not going to talk about it at all, but this is a sort of British ancestry in which these are a bunch of European groups. They're coloured according to sort of different countries. And this is what a British population group looks like, and these are the European groups that we think it looks like. And the point is they're all kind of almost identical. If you look carefully, you can see there's slight differences between them in terms of like the amount of blue and amount of green they copy, but they're very, very similar. But the point is that we can model the British successfully as being a mix of these various different groups. It's like 30% of this one, 10% of this, and we show actually that this makes sense. I'm not doing it. So the point is, you don't need, all of this assignment is kind of almost completely inaccurate, but we have whole genomes worth of it. So based on all of those genomes, we can see what it constitutes very well, and it doesn't, the painting is nowhere accurate, So that's a kind of just a statistical message in some sense, which I think is very relevant to the point of this workshop. So that's kind of so now I'm going to give my actual talk, which is and sort of if we're going to do metagenomics, we're mainly talking about bacteria and archaea, and the idea is that our kind of how we should be viewing metagenomic data is simply in terms of the way that bacteria evolve. And most of the most reliable and simple information that we have about how bacteria evolve comes from taking isolates and sequencing them. Okay, so I think, so what I sort of do in this talk, I'll kind of talk about data sets which are pre-metagenomic in some sense, and just say, well, the challenge is how much of this can we infer from metagenomic data, and which parts are harder. That's just the idea. So I'm going to talk about not quite a metagenomic experiment, which is done with these people here. It's actually China CDC, and it's a really very simple experiment indeed. Actually, they've taken 18 Helicobacter pylori, they've picked them from the same biopsy, they've single colony isolated them, and then they've genome sequenced them. Okay, so this is a really very simple. So Helicobacter pylori live in the stomach. There aren't very many bacteria that live in the stomach. I don't think people know of any others actually. So you have 18 strains of these bacteria, okay, and then we whole genome sequence, and then this is just a neighbor joining tree based on the sequences, okay. And then the first thing that you can say is that first of all, there are two clades, okay, and what we know about Helicobacter pylori, if you just choose random strains from the population, so they live in the human stomach and nowhere else as far as we know. If you take random strains from the same population, you'll typically see about 2% sequence diversity between them, or 2 or 3%, depending on exactly where you are. Okay, so there's several percent. Okay, and the genetic distance between these two clades is typical of the genetic distance between unrelated Helicobacter pylori that you see in the same population. Okay, so there's a kind of very simple inference from that, which is that this person's stomach has been colonized by at least 2 different bacteria from the same population. That's a kind of very simple, so 2 infections. Okay, and if you have whole genome sequences, you can do a bit more. We have this piece of software called Clonal Frame, and what Clonal Frame does is to estimate a phylogeny, taking into account that some of the changes happen through recombination events that change many nucleotides at once. Okay, and in this data we get import size of about 1,000 base pairs. Okay, and that's similar to what has been looked at in other. There's some variability in this, but short genetic fragments. Okay, so we get these 2 clades, and based on other data, we can. So this is the data is calibrated according to the number of mutation and recombination events. Essentially calibrated by the number of mutation events. Okay, and you can see that within this biopsy, so one clade, the most recent common ancestor, is about 4 years old, and the other one it's about 3 years old. Okay, if these calibrations are accurate. Okay, it's actually a little bit challenging to calibrate sometimes the bacteria, but we've done that if the calibration is right. Okay, and the second thing that you can see, so all 18 strains are different. Okay, so that's kind of slightly remarkable, and also there are actually recombination events. Okay, so they have imported strains on every single branch of the tree. Okay, Helicobacter pylori amongst bacteria is kind of famous for how much it recombines. It's when they first kind of did homoplasy tests, they kind of noticed that even very closely linked nucleotides were often in linkage equilibrium or close to linkage equilibrium. So, and this is the kind of the process that we have on every single branch of this tree. You know, although some of these strains are only sort of months separated, we have recombination events on every branch. Okay, so the kind of the most is 61 events on this branch, and it varies a bit. But every single branch has recombination events. Okay, so that's the next thing we can do. And we can actually look at where the sources of these recombination events are. Okay, what we've done is, so this is the clade A, the smaller clade at the top, and we were asking for the recombination fragment, how far is it from the best match amongst the other strains in that clade and amongst the best match strains in the other clade. Okay, and the kind of important thing that you can see along here and along here is there are many strains which are nearly identical to the sequence from the other clade. Okay, so what we can say is that this is recombination between, so these 2 strains are sitting in the stomach, and what we show is that they are exchanging genes with each other. Okay, so in some sense that's what we can do. Okay, so there are others which are kind of similar to other strains in their own clade, but there's also a bunch of strains which are similar to neither. Okay, and maybe these are recombination events with something else that we haven't sampled, but that is in the strains. Okay, so I just show you a few, 2 or 4 examples of different gene trees for different strains. And these are the gene trees. So this is a kind of very straightforward tree here. So we have the 2 clades coloured in red and blue. Okay, and you can see that there's a single strain in clade B which seems to have picked up a copy from single strafe on this clade which has picked up a copy from the other clade. Okay, that's kind of, or was it the other way around? Getting confused now. Okay, here there are actually two different apparent imports. So this is an example. So first thing is that most of these strains seem to have a single sequence. Okay, so maybe like the common ancestor has recombined, and I'll show you evidence of that later. Okay, but here we have a strain which really has something completely different. Okay, so that's another interesting case, and this is kind of the most interesting gene. Okay, so these strains are typically very similar to each other, but at this gene, almost every sequence is different. Okay, which is kind of spectacular, and also it's kind of pretty mixed up. You can't. So that's why I don't think you can really, based on these sequences, you can't really infer a history because it's so complicated. And secondly, there's clearly been lots of exchange between the two lineages. Okay, and actually this is a phage-related protein. Okay, so what it shows, so we don't really know, so the bacteria do uptake naked DNA. We don't know in Helicobacter pylori how important phage infection is, but this seems to be a completely active, ongoing infection which is very rapidly cycling between these two strains. Okay, that's what it shows, and it's kind of a really very dramatic exchange of DNA. So you can kind of see all of this. But I kind of, if you think about metagenomic data, you know, inferring this kind of, and the kind of the bubble and the challenge, inferring these kind of things and making a history is very, very difficult. Okay.
I would have thought that there's not stuff happening dramatically in D because they've all had time to mutate away from each other, whereas up there maybe.
No, but these are from the same organisms that have these genes. It's the same 18 organisms. Okay, most genes look like this, or they have no variation in them at all.
Yeah, something like A, where there's no red and blue interspersed, and that would be.
So that's a proof, but there is red and blue. There is red and blue interspersed here. Look, there are four blues here.
But they've got long edges, so that there's more time's gone by. Like number 17, it's had.
No. The time is the time. The time is the time of the strains carry the gene. So the time that has gone by is only the 3 years that this strain. Each lineage has been evolving because we have this clonal background. So we know that the strains which carry this phage only have common ancestors 4 years ago. Okay, 3 or 4 years ago. So all of these variation has been introduced within 3 or 4 years. Now the history of the phage may be different. Okay, but all of these different variants have been swapped around and moved into these strains within 3 or 4 years. Okay, the different so it's kind of that's kind of what clonal background gives you in some sense. Is that it tells you that. Yeah, it doesn't tell you the evolutionary history of all these different variants because they could have come in from elsewhere. But what it does show is that it has been, so, you know, there's been at least 1, 2, 3 swaps between these lineages. You know, there's been several events at this gene. Okay, and yeah.
Was this an experimental setup, or was this
just
observed from one individual?
This is just, they've taken a single biopsy and they've just picked 18 strains and they have sequenced them all. So it's kind of why I thought this is very relevant. This is kind of metagenomics, but not quite metagenomics because they just sequence the genomes. You know, it's kind of, you know, and ideally you'd like to be able to infer which genes are recombining and all of that, but it's kind of challenging. Okay, but it's kind of these are the events that you see. Okay, and just this is the kind of the most. This slide is a bit blurry. I apologise. This is the kind of the most detailed thing you can possibly see. So this is a rather typical recombination event. This B4 here has picked up what the A has. Okay, but actually you can see that there is a single nucleotide within this imported fragment where it has kept its ancestral sequence. So there's been a kind of the recombination event, and this has been observed in vitro before, but during the recombination process, it's actually kept a little bit of the ancestral sequence in there. It's not a kind of a perfect copy, but there's this kind of been spliced in somehow during the copying process. So it's a kind of, it's just an interesting, you know, even where you have recombination, it's a very, you know, it's sometimes a subtle process. Okay, it's actually, you know, that's actually a mixed sequence of the two ancestors. Okay, it's kind of recombination within a recombination. Okay, so you know, so there we are. Okay, and then the other kind of thing that you can do, of course, is you can ask which genes. So these genes have been swapping genes. I've told you that there's this very interesting phage which has swapped multiple times. You can just ask what classes of proteins are exchanged more and what less, and so it's a lot of genes, so you expect it to be to some extent neutral, and you do see all different genes are being recombined at fairly high frequency, which we know. So what we also knew from the hot protein, which is an outer membrane protein, this seems to be particularly, we replicate the observation, this seems to be particularly frequently exchanged. Okay. And then the final thing, so I said that I told you that the common ancestor of these strains was 2 years and 4 years for these two strains, or 3 or 4 years or something. But you can actually, so what clonal frame does is actually infers the ancestral sequence. Okay, and so we can look at the ancestral sequences of the two clades and ask how similar they are to each other. Okay, now if you look at typical Helicobacter pylori, so I says in this population, this Chinese population, you kind of see 2% divergence with some range. Okay, but there's about slightly over 10% of their two genes, they're identical. Okay, and so the explanation for that is that actually the common ancestors of the two strains have already been present in the same stomach and recombining for some period of time before we sequenced these strains. Okay, so essentially, and it's about 10%, and that's actually so that implies that's probably about 12 years, assuming that the rate was the same as it is now. That would be about 12 years of evolution in the same stomach in order to get that. I mean, there's a lot of assumptions that go into that, but yeah.
What about just gene transfer? Maybe that one copy is so good that it just gets transferred into everybody.
Yes. So sure, there is some selection, and of course, most of the natural selection will be on the things that happen at the beginning. So, but we can't tell. But this, you know, it's a large proportion of the genome. So, well, we don't know how much it's selected. I mean, yeah, we can't answer that question. We'd like to be able to answer. I mean, we can start to, if we get this kind of data from a lot of individuals, then you can start to address that question, I think. So, okay, so there we are. Okay, so that's a kind of, you know, that's a kind of metagenomic scenario where we haven't done metagenomics. I sort of asked the questions. So, you know, the first question, in some sense, I think, what you want to know, I mean, in very complex systems, the answer may be very many, but the kind of foundational question is, you know, how many infections, how many different organisms have come into that and established themselves, and then what proportion of the population? That's the kind of the community composition question. Okay. Then we have the microevolutionary questions, okay, which are rather challenging to address using metagenomics. So Jack will talk about this after lunch. What diversion has each infection undergone, and you can also ask about functional effective diversification. And I kind of say that there is a heroic question. You can even see infections that aren't there anymore because we've seen some genes which don't come from, so they may be there, we haven't sequenced very many strains, but there are other Helicobacter that have left their impression in these genomes, so you can even answer a little bit about that. So this kind of, that's just a set of questions really, because my talk was a question, so I sort of put a lot of questions in my talk. So we kind of had a very similar list actually in Rosalind's talk, but it's worth, so then we can talk about the ecological questions, and I kind of think the challenge of metagenomics is to really make it address these kind of questions rather than being descriptive. I think that's the great challenge in the field now, and that's what we should be doing, so I'll stand up and say it. And the ecological questions, which we sort of had just heard, so we want to know, you know, these two strains, they're in some sense compatible with each other, they've been competing for a long time. Why do they do, so why do they thrive in the presence of each other? You know, why is it these two lineages which have succeeded rather than any other? You know, when does competitive exclusion occur? I think these are kind of ecological questions, and I'll show you some data about succession soon. Why does that happen? And then there's kind of questions about transmission as well. Okay, and then so along with that, then strains when they arrive in their environment, they have come from somewhere else. Somewhere else will always be in some way different. Okay, so how much is genetic change responsible for allowing, you know, these strains, these Helicobacter are recombining like crazy. You know, that's a kind of insane amount of genetic exchange on some level. What benefit actually are they gaining from this in terms of it? To what extent is this important for transfer between different environments? Okay, so that's, you know, we should be making our methods address these questions, I think. Okay, so I want to show you, so this work is not mine at all, it's from Frank Coles in Oxford. So this is a broiler, a chicken broiler enclosure. These are not broiler chickens, they're pet chickens for that. This is another not quite metagenomic experiment done by Fran, and she published the first thing here. And so what if you look in the broiler's chickens, then for the first few weeks there is no Campylobacter. So Campylobacter is of interest because it causes gastroenteritis in humans. It upsets chickens a little bit, probably not, but you'll find it in most farm chickens. Okay, what happens when they arrive? They're clean of Campylobacter. You get kind of first infection, and then just of a single strain, it seems, and the diversity picks up. Okay, and then it kind of, you reach a kind of stable state. Okay, most of this has happened before they're sort of let outside into the environment, just the way it happens. Okay, so this is the data, and this has been followed for a little bit over a year, and then something nasty happens to the chickens, I think. Okay, so this is the data. So the data that you have is this is pre-genomic data here. Genomic data would do quite a bit more for you, but this is MLST data, so they've sequenced 7 gene fragments. Okay, and what this is good for is if you're identical in all 7 genes, then these strains share a recent common ancestor. Okay, although some of these sequence type are common enough in the population that they could still have come in separately more than once into the chicken coop, but it's kind of more often than not, if 2 strains are identical sequence type, then maybe they're the same infection. Okay, although genomic data would tell you that for sure, so you can now address that question much more accurately. Okay, so this is what the situation looks like with based on isolate sequencing. These are for just a selection of the chickens. Okay, if there's no sampling, then it's just a box. If it's an empty box, then it's just culture negative when she's grown it. And what you can see is that culture collection is a somewhat tricky process. So sometimes you clearly see that the same strain has carried on the same infection, but you also see lots of examples of colour changes and everything. So it's quite. This is the kind of situation where you kind of clearly think you would. If you could resolve these lineages using metagenomics, then metagenomics would kind of be superior, because you could see, you might have a better idea of the what's overall in this chicken at any one time. So it would be good to be able to do that. These lineages, from a metagenomic point of view, is pretty similar to each other, but if you have genome data, you should be able to distinguish them. If you view the whole breeder flock as a single organism, then you get something which is more like metagenomic data in some sense. And what you see when you do this with this resolution is that there's actually lots and lots of succession. So the very first strain that came in made up the whole population, was disappeared, gone within a month. Okay, so it had its time and then it disappeared, and there were kind of several. There's a lot of succession going on here. So this genotype is very successful for a while and then kind of fades out, and you get this. So there's a lot of succession in this system. Okay, and you get a genotype which does well for a while, disappears, and that kind of thing. So, we'd like to know, what is responsible for all of these patterns and this kind of thing. And I think that's a kind of classic metagenomic setting in some sense. So, but with whole genome data. But in some sense, what I kind of want to say is that the end goal that we should have in mind is for metagenomics is to really do this at the level of individual infections, in some sense. So you can say this is one lineage which has established itself, grown and shrunk, and grown and shrunk, and I think that is going to add, from the point of view of most metagenomics, all of these strains are kind of the same. Okay, but there are a lot of interesting stories in following individual lineages, I think, in terms of the biology of these things. Okay, so now I'm going to kind of get more and more bacterial population genetics. So in talking about, so you have populations of bacteria, and to kind of understand the dynamics, you sort of want to know what actually are the set of things when you sample from an environment. When an environment samples from outside, what actually is it sampling from? Okay, what are the range of things that could have infected that environment, so that we can then sort of compare that with what we actually see. Okay, and I'm going to talk about one example of this. It's Vibrio parahemolyticus. It's an ocean bacteria, so kind of ocean is great in some sense because this is an enormous sort of apparently homogeneous environment, so it's kind of very interesting place to look at metagenomic questions. So what's kind of interesting about Vibrio parahemolyticus is that it seems to be, so this is genome sequences from about 150 strains. Okay, now these are not at all, so this is an ocean bacteria, but they're not at all sampled randomly, and the reason they're not sampled randomly, this causes gastroenteritis in humans, and a large proportion of the strains are clinical. Okay, so and others are from seafood, okay, or from the environment. Okay, and you can see in particular there are certain lineages. This lineage here, which is called CG1, this has been responsible for a large pandemic worldwide. Okay, so people have collected it, and all of these strains are really very similar to each other. Okay, and that's kind of a clonal lineage within the population. Okay, and there are a bunch of other lineages which together are responsible for a large portion of human disease. Okay, but so when you go outside these lineages, what is striking is that you get this kind of perfect radial bush in your tree. Okay, now what this radial bush means, okay, is a kind of diagnostic for the fact that the strains are essentially freely recombining genes with each other. So if you look, for example, in humans or other eukaryotes where you have sexual reproduction, and you look in a population, everyone is kind of equidistant, genetically almost exactly equidistant from everyone else. Okay, because it's a kind of random process. I mean, if you have a bit of geography, then you'll see slight differentiation, but there's a kind of essentially a random swapping of genes. Yeah? So all of these, so there are a very large number of very weakly supported branches in this neighbor joining tree. Okay, so this isn't true for all bacteria, and it's not completely true here because, as I said, there are clonal lineages where you see less variation. Okay, but we kind of have this point, and so the way this happens, it's kind of the way to think about the kind of ongoing process of bacterial evolution that causes this is that, so these are all the strains in CG1, this clonal group which has caused a lot of disease. Okay, now there's one hotspot of diversity, and in fact this is for the, forgetting what its name is now, the capsule which is expressed on the surface. Anyway, so there's a single island, and this actually recombines a lot. Okay, there are there are 3 recombination events of this entire island just in the history of CG1. Okay, but there are no other big recombination events across the entire strain. There's one part of the genome where recombination happens a lot. Okay. However, if you then add a strain which is further diverged in terms of time, you then see recombination events which have introduced diversity at several different places. Okay. So if we go, so that was CG. So first of all, we looked at all the SNP diversity within CG1. Then we added this strain here. Okay. So these strains here, they're related. They kind of share, but they're clearly sort of different from the pool as a whole. Okay. And what you can see is that sort of large proportion of the genome, you see lots of SNPs, but actually there are kind of islands where there has been no recombination. Okay, so if you add that diversity, so you can see, so those strains share a clonal frame by direct descent at a sort of a proportion, maybe a third of their genome. Okay, and then if you just add one more unrelated strain from the tree, then you see that there's high diversity across the entire genome. Okay, so there's a kind of, if you take 2 unrelated strains, you'll see a pattern of diversity that looks like this. So there's just a high level of SNPs right across the genome, separating all of the trains. And that's a kind of, this is kind of what a sexually recombining population looks like. Okay, so you know, bacteria, they sort of look like a sexual population, but you see clones and you kind of see half clones, okay, and all the way along. And that's what you see. That's what you see in bacteria. So. There is actually differentiation in Vibrio parahemolyticus though, and it's quite interesting. We found, so here what we've done is we've made the ancestry of each isolate into all of these others using the chromosome painting software, which I mentioned but won't describe. But what actually you see, so these are kind of signatures of clonal groups, but what actually happens, so when you take one individual from each of these clonal groups, and one individual from these ones, you still see there are these strains, the US population strains, which are sort of different from the Asian population strains. They copy, they donate less. The Asian strains don't want to use chunks from the American population. They're kind of differentiated. Okay, and it's about an FST of 0.07 between these US population strains and the Asian ones. Okay, so this is quite interesting. It says that the entire Asian Ocean from sort of China to Malaysia, or I can't remember the exact distance, is a single gene pool. Okay, there's no detectable differentiation, but if you go to the US coast, then you kind of see a 7% FST. Okay, but for example, the pandemic strain, which I showed you, that has successfully gone from one continent to another, so it appears everywhere. So what it seems like is that there is actually geographical barriers to exchange for these bacteria, okay, but it's not absolute, okay, but there has been enough exchange that there is genome-wide differentiation, kind of similar, so a bit more than it's similar level to you see in humans between continents, I suppose. Okay, so there we are. Okay, now, what we did then, so we got all these bacteria, so actually it kind of suggests that these bacteria are kind of a single gene pool, that the geographic movement, at least within the Asian Ocean, these bacteria are kind of, they're all drawing from one gene pool and they're kind of random associations from that gene pool. Okay, so now, so we kind of, you know, it's slightly interesting. If they're doing, one might imagine that in different places they have different, not just geographic places, but in terms of how saline it is and all these other things. They might be doing different functions, in which case you'd expect to see non-random associations between different loci in the genome. Okay, so what we did here is we took 53 genome-wide unrelated isolates, okay, and then we asked which parts of the genome showed strong associations. Okay, so if they were kind of different functional groups, then you'd expect to see non-random associations. Okay, and so this is a QQ plot. Okay, so we've done a Fisher's exact test of association between all pairs of SNPs, essentially. Okay, now the red line shows all all of the pairs of SNPs in the genome. Okay, and you see lots and lots and lots of non-random associations. Okay, however, it turns out that almost all of them are between SNPs that are, most of them are actually within 2 kb. There's a few up to 3 kb, which are quite interesting, but these are all SNPs which are on the same part of the chromosome. Okay, and so this gives this nice curve of associations. Okay, if you look at SNPs which are more than 3 kb apart, there's actually only one set of genes. So I don't think we have very much statistical power here, but there's only one set of genes which, based on 53, there's one set of genes which we see a non-random association, and that's, I think it's involving the LuxR gene and a promoter somewhere else in the genome. I think it's real, but there's only one non-random association. And it's kind of, in some sense, this is slightly worrying for metagenomics, okay? Because it says that when you see strains in your sample and you think you have a gene which is associated, what it says is that most of all of the kind of functional association, a large proportion of what you think is functional association can simply be due to linkage. Okay, so we've removed all the linkage due to clonality. Okay, but still all of the linkage that we see is just due to co-inheritance of DNA. Okay, it's just all of this is due to co-inheritance of SNPs together. So there's very little functional association outside that. So it's kind of worth bearing in mind that the bacteria come in as they are, as a single entity, and they don't have a time to kind of fully mold themselves to the environment. So it's kind of, you're getting the whole bacteria or nothing in some sense. That's a kind of interesting observation. Okay, so that's Vibrio that's in some sense a relatively simple population structure because the strains are from a nice random mating pool. Okay, but other populations are more complicated. This is the ever-growing lab of Sam Shepard, and what we have here is so this is Campylobacter jejuni, and this is a neighbor joining tree of the strains. Okay, and what you can say, so they've been color coded. So they live in chickens, which we've seen. They also live in agricultural ruminants, and they live in wild birds. Okay, and what you can see from this tree, so there are lineages which are sort of well recognized to infect single hosts, or I mean they're pretty much the same. So this is kind of a big group of chicken associated lineages. So you can imagine, a Campylobacter sort of came into a chicken here, and it found itself very at home, and it's been adapted to chickens. Okay, and you can say the same thing here with ruminants. Okay, so wild birds, which are the presumed sort of native hosts of Campylobacter, they tend to have their own lineages. You do find 45 complex in them. They tend to have, we haven't, people don't care enough about the actual natural world, but from what we do have, they have their own lineages. Okay, but the kind of most interesting thing is that you get these complexes. Here they are 21 and 45 complex, which are multi-host. Okay, and this one is particular. It's not particularly frequent in wild birds, but it's mixed chicken and ruminants. Okay, and I kind of point out that I'm not really a biologist, as we heard, but I kind of, which is related. It's an interesting question that I will leave for your judgment. Especially that they're living in here and here and here, and you sort of have to decide. So so agriculture is a really weird place because it's clearly its own thing, in some way. Okay, and so you can zoom in on the microevolution within these host generalist lineages, and you can see how often actually are they switching. This is actually from another species, Campylobacter coli, but you kind of see the same thing. And what the tree makes clear is that they're switching hosts quite a lot, actually. So it's kind of clearly, if you can infect multiple hosts, then it offers transmission opportunities, and if transmission is limiting, then that's going to help you. So, but there are a lot of differences between the innards of a chicken and a cow, actually. So it differs in immune system, it differs in temperature, it differs in what they eat. So it's to some extent how it happens like this is something of a mystery. What we did to start to investigate this, so because we can see that they switch host often, then there's a lot of potential adaptation events. Okay, and particularly because when you go into a new host, they can pick up genes from other strains in that host. So there's a lot of opportunity for kind of on-the-fly adaptation, so we thought we'd see if it happens. What we did is we did a word association study. We just took 30 base pair words in the genome, and we asked if they were present or absent in different strains. Okay, and we just did that for all 30 base pair words, and by that process we have a hope of picking up either single nucleotide changes or recombination events or gene gains and losses. So we kind of it might pick up everything, it won't pick up quite everything. And what we found 10 regions. So we did this in a single clonal complex, and we realized that because there isn't much variation in the complex and it switched hosts several times, then we should, if there has been a set of changes which happen frequently, we'd have a lot of power to pick it up because the general background is relatively homogeneous. And we found 10 regions where there was some evidence for host association. But then we tested it in the other lineages of Campylobacter, and only one of the regions really, only one of them replicated. And that also was the one which had 76% of the words, so we got about I think there are about 7,000 words in total. I think it was in region 3. And what this turns out to encode for is it's a gene presence or absence trait, which we got so many words, and it turns out it's the vitamin B12 biosynthesis pathway. In these two complexes, so in one of the host generalist complexes, it's always present. Okay, but in the other, it has been lost or gained 2 or 3 times, so yeah, I don't think we can really be sure which. And so vitamin B12 is necessary if it's necessary for the bacteria, and it occurs it is very rare it's very low frequency in grass. Okay, so cows eat grass and chickens eat something else, grain. So it seems to be that to thrive in a chicken you need to have this. And that's the kind of thing. And interestingly, actually, there's also an antibiotic gene which is right next door to this. Well, it's not proven it's antibiotic resistance gene. The interesting thing is that that's present in very large proportion of agricultural strains, and all of the sequences are identical. Okay, so this gene has spread within agriculture very, very recently, and it is right next to, I think it's this gene here, and it is right next to this vitamin B5 pathway. So somehow this genome has put these kind of agricultural adaptation genes, so the pan-BCD is also very much more common in the agricultural strains, even from chickens, than it is from them in wild birds. So we kind of have this agricultural adaptation complex, okay? And so far, what it seems to be vitamin biosynthesis and antibiotic resistance, possibly. But it's interesting that they occur together. And yes, I think I should stop.
Thank you very much. Open for questions.
So this was a phylogenetics conference. I don't think you've been getting away with just using neighbor Johnny. So did you look at other?
Wait, which? So I use clonal frame, which is this. So I don't think any phylogenetic methods are appropriate.
Okay.
So we have this clonal frame, which is evolution through, and we infer ancestral traits with mutation and recombination. Okay, so that is a more appropriate tool. Okay, I don't believe that other distance-based methods are. I don't think that likelihood-based a better model of this data than neighbor joining, for example.
I got interested in your common to mixture modeling, and I thought that a few days ago when we had discussed this, I had understood you, but I got a different message today. So I just want to check that I understood the right thing. So you are suggesting that we should be modeling complex communities of microorganisms as an aggregate organism made up of several different components, and not worry about treating the different organisms that populate the community as things that made up the components of the mixture, and not worry about binning the different organisms by components or just
no. So there will be there is a bidding state at the beginning, okay. But my point is that the bidding doesn't need to be. You shouldn't aim for very accurate bidding. What you should aim for bidding, which you can kind of model in some sense what its errors is, and that you can model. So given that you have a strain, you can ask if you put a real genome in there, you can see what there will be some proportion of incorrect binning. Okay. Now I don't think that is a problem at all. Okay. When I paint a European genome with a worldwide sample, some of it will come from Africa. Okay, but that doesn't lead me to conclude it's African, because all European individuals look like that. Okay, so the point is that if you have a good mixture model of how your binning works, okay, then your binning, and you kind of have to think about a kind of binning technique which makes that work. So if you have a good statistical model of how that works, then your binning can be quite noisy, and you can still recover perfect information from it. And the example of the peopling of the British Isles is that people in Northern Europe, they're all virtually identical, okay. And if I say who is your closest ancestor in the room, it's just kind of random, okay. But there is, on average, there is a little bit of a signal that Norwegian people copy a little bit from Norwegians, and you can make quite detailed inference based on that. So I just think that the focus should be away from accurate individual classification and towards methods that allow you to aggregate a lot of noisy information. That's the kind of, and that's what you're going to say in your talk in some sense, is it? I apologize for.
I have a question. If I understood it right, you were saying that in your initial study, you assumed that there were two strains initially causing an infection, maybe because the most recent common ancestor of some genes was before the time of infection, or why did you base that?
Oh, sorry. So
When you were
using our better than phylogenetic method, because it takes into account recombination. So using this method called clonal frame, so what we do is we reconstruct a tree and the common ancestor, so we reconstruct the ancestral sequences in the same way as phylogenetics, but also sort of numbering the recombination events. Okay, but anyway, the point is that according to our calibration, okay, this lineage had a common ancestor 4 years ago, and this one had it 3 years ago. Okay, so we've taken these 10 strains from these two samples. Okay, but then you can ask, what did the ancestor of each of these two strains look like? Okay, now we have a model that if you take unrelated Helicobacter from the same population, then at most genes they'll be 2% apart because there's this random recombination. Okay. Anyway, what happens when you look at the distribution of nucleotide divergence of these two ancestor sequences is that there's a 10% of their genome falls in the like no diversity bin, okay, which is kind of unexpected if you if they're unrelated. Okay, now the explanation that we have, and it's just a model, is that originally the curve looked like this, okay, and what's happened is that while the ancestors of these strains were in the same stomach, they have been exchanging DNA, okay, in the same way that they've been exchanging DNA since, okay, but because we don't have any survivors from back that time, we we can't see which genes it is, but we can see that at 10% of it they have converged by recombination, these ancestors. That's the logic.
Time for one more question. Maybe I can ask one. So it seems there's a story about survival here, and it's not necessarily about the strains. It may well be about the genes. Can you comment on that, please?
So it's both, of course. So the gene needs an organism in which to survive, and the organisms need genes to help them survive. But of course the genes, in these recombining bacterial species, genes will only survive if they transfer successfully. So most of the genome of these bacteria has been acquired within the last 100 years. Okay, the cellular body is, I mean, it's the experiment that Craig Venter did, where he took out the whole genome and put it back in again, or whatever. These bacteria are doing that, but they're just not doing it. They're doing it over a time period of decades. Okay, depending on the bacteria. Okay, so and it's actually, so we have this thing in Vibrio I haven't presented, but if you look at the age of the effective population size of the strains is completely different from the effective population size of the genes inside it. So there's a kind of cellular story, and there's also a gene by gene story, and they do tell you different things, actually. So.
Good. Well, with that, let's conclude this session. Thank you very much, Daniel.